Hi, my name's Sam. I'm a postdoc here at AgResearch, and today I'm going to tell you a little bit about a trial that we ran as part of the Capturing the Value of Red Meat MBAE program. It was a nice trial, which was a collaboration between the University of Otago, Callaghan Innovation, AgResearch, and Scott Technologies. And it's all around how we can try and predict meat quality parameters using non-destructive sensors. My contact details will be at the end of the talk, so feel free to send me any questions. Okay, so we are interested in trying to determine the meat quality parameters without having to use chemical or destructive sensors. And how we're going to do that is a technique called Multiblock PLS modeling, which allows us to combine the data from a variety of different sensors to try and predict their quality outcomes. Okay, so why do we care about meat quality? This is probably preaching to the choir, but New Zealand beef and lamb exports are forecast to exceed $9 billion in the year 2019 to 2020. So that's 4.6 billion for beef exports and 3.5 billion for lamb and mutton, and the remainder being for New Zealand for the co-products, according to the Beef and Lamb New Zealand mid-season report. Fortunately for us, 90% of that that meat is exported to countries willing to pay a premium for quality meat, and so there's a need for a fast, accurate, and non-destructive estimation of key quality parameters, including intramuscular fat, pH, tenderness, and many more. But we're particularly interested in intramuscular fat and pH for now. So we need a system that can determine these things without having to cut up meat and send it off to a lab or anything like that. And there are going to be two key outcomes. One, of course, is guaranteed quality for consumers. So we're sending meat out, which has a stamp of approval of however much intramuscular fat, whatever pH that each individual market may be interested in. And of course, we're able to then send that information back to farmers, which may help influence their feed that they choose to use, their breeding flocks, whatever. So information is really important for the farmers as well. So we designed with all of our collaborators this robotic sensing platform, which really allows us to take consistent data across many sensors on the same sample on the same day, which can really allow us to make some key decisions around what sensors may be able to be implemented in a pilot plant or a meat processing plant. So we used we designed this robotic sensing platform. At the center is a robotic arm, which controls the lighting for our sensors and initiates the different sensors that we're using as well via these Arduino microcontrollers. We used 3 different hyperspectral imaging cameras, a line scan camera, which requires the robot to translate the sample underneath the camera. We used 2 snapshot cameras at different wavelength ranges, an NIR spectrometer, so that's doesn't collect an image, just collects a single spectrum, but has a much wider range than the hyperspectral cameras, and also a Raman spectrometer, which, for those who aren't familiar, excites the sample using a high-intensity laser, and the excited sample gives you back a different kind of spectrum than the NIR spectrum, and maybe some complementary information there. And we also have offline sensors, another NIR spectrometer. The group from Scott was doing nuclear magnetic resonance relaxometry. And then we had another Raman spectrometer as well. The samples that we used took a lot of thinking about. So we wanted to have as wide a range of intramuscular fat and pH as possible, so that we make sure that when we're conducting this experiment, that we're seeing everything that we the range that we would expect to see in any particular pilot plant around New Zealand. So we chose to use 52 Wagyu Friesian crosses. This was to ensure that we get wide ranges of intramuscular fat. We used 130 bull samples, and 65 of those we cold shortened in order to get a varying tenderness profile as well. And so what we did was we took these samples of short loin, we cut them into cross sections, and we laid them on a tray like this, which was placed into the robotic platform, robotic sensing platform, and was carried around to our sensors. So we had a short loin. It was cut in half, and then the two halves were then placed open on the sample on the tray. Okay, so here we can really see why we chose these samples. The pH was measured immediately before scanning with all the sensors. We can see that the bull samples had a very wide range of pH from 5.4 all the way up to nearly 0.7. But the Wagyu, of course, had a very narrow range of pH. So the combined data set had the range that we were interested in seeing. Conversely, the Wagyu beef had a very wide range of intramuscular fat, as opposed to the bull, which had very low and very narrow ranges of iron, all between 2.5%. But combining those samples, we get a nice range for modeling later on. Okay, so here's the robotic platform in action. What you're going to see is in the center here is our robot, which just out of view is the sample on the robotic platform. The first thing it's going to do is it's going to move over to the contour mapping phase. We need to measure the height, so that the height of the sample, which changes a little bit each time depending on the cutting, to optimize the imaging of the spectrometers. Going to go there first, and it's going to move over to our NIR and snapshot system, into the line scan system, then to the Raman sensor. So, okay, so there's your contour mapping. Took an image of the surface of the sample. NIR spectroscopy, and there are two snapshot cameras there. And so here's the line scan camera, and it obviously requires translation with the robotic arm. So that's what you saw there. The robotic arm also closes up that light, because the Raman spectrometer is very sensitive to light leakage, so closes up that pneumatic sensor. So hopefully there we've collected some really nice consistent data across many sensors, and we repeat that 364 times, or however many it was, in order to build up this data set. So how are we going to analyze this data? What we use is this technique called multiblock partial least squares modeling. It's a machine learning approach which allows you to treat the data from each one of those sensors as a block. And when you have multiple blocks from each of these sensors, then you can use the system to predict the combination of all these sensors to predict a single output, which for us is going to be either the intramuscular fat or pH. And we stack up these blocks with the data from all of the different samples, make sure they're all aligned so that you don't get any errors, yeah, and so there's obviously a lot going on in the background there, but that's going to be our approach. Okay, so here are the results. On the right-hand side, you see the predictions of IMF using Multiblock PLS, using all of the sensor data, and here are performance metrics. A high R-square, but that's to be expected because we had such a large range when we include the Wagyu samples. When we look at each of these samples, when we look at each of the sensors individually, the blue plot here is the R-square, and the red is the standard error of prediction. So what we can see is that actually all the sensors combined is not as good as just using the NIR and line scan hyperspectral cameras when trying to predict IMF. And so the other sensors, not only do they not have useful information, but it's looking like for this particular parameter that we're being that they are just muddying the issue about predicting IMF. We actually see something similar with when we're trying to predict pH. Again, the line scan camera works the best, and the two NIR spectrometers and cameras and snapshot cameras are all approximately. So it's a shame that the nuclear magnetic resonance and the Rugaku here is the Raman sensor did not predict these parameters. However, there's a lot of evidence that they can predict other parameters such as water holding capacity and that sort of thing. So so there's a potential there for all of these sensors to be used to predict different parameters, right? And we've only so far looked at pH and IMF. Okay, so we have a lot of data from a lot of different sensors, and now we're starting to think about, well, not only do these sensors cost different amounts and have different wavelength ranges and different implementations, that sort of thing, what sort of combinations can we use to start getting some improved results? So I'm just going to talk about a case study that we had when we're looking at the snapshot cameras as opposed to the line scan camera. So as you saw previously, the line scan camera gave us the best results. However, it turns out that it's quite annoying to have to translate the samples like that. And it's slow to do that. And so there are issues around actually trying to implement this in a real, in a working meat plant. What we do have is these two snapshot cameras, which on their own didn't perform as well, but because they have they cover different wavelength ranges and have different number of bands, but are much faster at imaging, can we use the two cameras together in order to predict? And I'll just show you the results for pH, but in order to predict any of our parameters and overcome the limitations of the line scan. So here's a bit of a look at the predictions. So the snapshot in the visible range, this was the prediction curve. You can see that there's it's not nearly as tight as this line scan camera on the left-hand side there. And the NIR was even worse at predicting the pH. But like I say, because we have two different wavelengths, potentially these can be used with Multiblock PLS in order to get a better prediction. So we went ahead and did that, and what we found was that that the Multiblock PLS with the VIS and NIR data combined in general gave a really good prediction, except that we ended up with some large outliers, which were not present in the previous results. However, some of these, so this guy here and these guys down here, are outside of what we would ever expect to get biologically. So we can remove those. And so when we remove these outliers, we actually end up with a better prediction than we got just using the line scan camera. So we went from 0.9 to 0.91 and with a R square and with a slightly lower standard error prediction. So so a nice result, and hopefully we can repeat this with some of the other quality parameters that we are interested in. So in summary, we collected spectroscopic data on intact states with wide-ranging IMF and pH values. We've designed this robotic sensor platform for automatic and consistent measurements across all those sensors. And in a case study, we found that a couple of the snapshot cameras, which are more appropriate to be implemented in a meat processing plant, could be used together to get as good a performance as a camera that was not as easy to implement. And of course, this is just the beginning of the types of analysis we can do with these with this data set that we've created. And so I reach out to you. Is there anything that you would like us to have a look at? Thank you to all of the people that were involved in this project. Feel free to contact me with any questions you might have. Thank you.